Hello everyone, and welcome to phyloseminar.org, a project supported by the Society of Systematic Biologists. I think that we all appreciate that protein structure greatly determines the evolutionary trajectory of protein-coding genes, though it's rare for people to incorporate structure into molecular evolution studies. The reason that structure is typically not incorporated is because it's challenging. However, some courageous individuals are working to incorporate structural information into molecular evolution studies, and we have 3 of them for this phyloseminar theme. This will include Jesse Bloom today, then Klaus Wilke, April 20th, and Richard Goldstein, May 25th. As I mentioned, today we have a talk from Jesse Bloom. Jesse started out in chemistry, getting his BS in biochemistry from the University of Chicago, his master's in theoretical chemistry at Cambridge, then his PhD in chemistry at Caltech. During this process, he was progressively moving towards working on the impact mutations have on protein structure and function. He did his postdoc at Caltech with David Baltimore, where he did landmark work showing that stabilizing mutations are needed in influenza before resistance mutations can develop. He's continued to do very interesting work here at the Fred Hutch, where he recently made his promotion to associate member. Thanks for participating, Jesse, and I'm looking forward to your talk.
Thank you. It's a pleasure to get to give the Philo seminar today. For those of you watching remotely, we actually have 2 live audience members today. Eric, several live audience members, Eric and people from his group. So I'll report at the end of this seminar whether they're not only alive but still awake to anyone who is watching remotely. So I'm going to talk today about using new high-throughput experiments to inform phylogenetic models of substitution. And this is a theme that my group has been working on for the last 3 or 4 years. So just as way of background, because most of what I'm going to talk about and probably what the next 2 Philo seminars are going to talk about are the assumptions that go into phylogenetic inference, I think it's helpful to just take a step back for a minute and think about what we're really trying to do in phylogenetics. So in phylogenetic analysis of gene sequences, there's some forward evolutionary process that creates some set of gene sequences, some small subset of which we observe. And in phylogenetics, what we're really doing is we're making models of how these sequences evolve to allow us to make inferences about this forward evolutionary process, which we don't really observe in full detail. And there's a tremendous number of assumptions and simplifications that go into how we think about this forward evolutionary process. So if we were to, what would an actual accurate description of the forward evolutionary process be? Well, we imagine that there's gene sequences in each of the individual variants of the gene that we're studying, and each individual has some sequence, has some genome which contains this sequence, and there's some big set of these genomes and all the individuals that exist in the world. And conceptually, you can imagine the real forward model of how these things would evolve is you'd look at the whole set of the genomes that are in the world at some time, and there's some function which reflects the selection that acts on those, everything else that's going on in the universe, and then random effects due to things like quantum mechanics, and that's what determines how the genes evolve. And as you can imagine, this sort of, I guess, complete description includes all sorts of processes that we have no hope of actually modeling in phylogenetics. So, for example, none of the phylogenetic models that anyone even conceives of describe some asteroid coming from outer space and wiping out the dinosaurs, although that obviously had probably a lot of impact on sequence evolution over long time spans. So what are the actual assumptions that we do make when we look at this process? So there's a fairly standard set of simplifications that are used in phylogenetic inference. So first of all, we typically assume that whatever gene sequence we're looking at can be considered independent of the rest of the genome. So typically when we build phylogenetic trees of protein coding genes, we're doing that of individual genes, and we're not thinking about the larger context in which they occur. Second, we usually think of these sequences as evolving independent of sort of the structure of the populations in which they exist, so we're ignoring those effects. And third, we generally assume that this evolution is only a function of the mutations that occur and the selection that acts on these mutations. So we don't worry about these processes like asteroids wiping out the dinosaurs. So we have some sequence, and we think at some point in the future, we can describe the evolution of that sequence both in terms of the mutations that occur and then the selection that acts on those mutations. And so you might conceptualize this evolutionary process as something like this. You have some protein sequence, and at some unit time, there's a mutation that occurs in one of the variants, and we look at phylogenetics at what would happen after selection has either removed that mutation or that mutation has gone to fixation. And we're sort of thinking of the evolutionary process in this sort of simplified way. And then we generally make a lot of further assumptions, most of which I'm going to continue doing in the work that I'm going to describe here. So first of all, we usually only consider substitution mutations. So we're only going to be looking at substitutions in these genes. There are methods that look at indels, but they're not that common. And then another big assumption, and this sort of starts to get to the crux of what I'm talking about, is we typically assume that sites evolve independently. So that is, for each site in the gene, we think that that site is identically likely to have mutations. And actually, this is not a necessary assumption, but for the current frameworks, we assume that these sites change independently of each other. So we've taken this long gene sequence and we've partitioned it into a large number of different sites, which might be nucleotides, they might be codons, and they might be amino acids. And so this is an assumption that no one really thinks is true, because in fact, all of these collection of sites in these genes all form the same molecule, and selection is really acting on that full molecule. But the reason that we make these assumptions is it allows us to apply a very powerful statistical framework, which is at the heart of, I think, most phylogenetic methods that people use, and was sort of originally developed in large part by Joe Felsenstein. And that's the idea of calculating likelihoods of what we observe in phylogenetics, which is some set of sequences in terms of some phylogenetic tree, which tells us how these sequences are related, and some substitution model that tells us how likely each position in each of these sequences is to change from one identity to another identity at some unit of time. And really, this assumption that the sites change independently is pretty important for this whole calculation, the numerical calculations that are done here, to work in a feasible way. And probably everyone listening to this is really familiar with this framework. What people would typically do in this framework is you would have some substitution model, and you would take that to look at your sequences, and you'd either find the tree that maximizes this likelihood, or you'd put some sort of prior over these trees, and you'd look through a bunch of trees and try to estimate the posterior probability distribution over these trees. So an interesting, so first of all, in my talk, I'm going to talk entirely about this part, so these evolutionary or substitution models. That's the part of the phylogenetic analyses I'm going to be talking about. Everything else I'm going to do is entirely standard. And one interesting thing about this framework is although doing these calculations in the way that Felsenstein describes requires us to treat these sites independently, so it requires us to look at sort of calculate a likelihood for each one and multiply those up, nothing actually requires us to treat these sites identically. So that's why I've written this subscript R here, and what that means is there's some probability for each site R that it will change from amino acid X to amino acid Y. Despite the fact that this computational framework doesn't actually require us to treat these sites identically, the vast majority of substitution models that people use in practice assume that sites evolve identically. This should say assume that sites evolve identically in addition to independently. So the vast majority of substitution models assume that this term right here can be written like this, even though that's not a requirement for any of these computations to work. And people in phylogenetics, including probably a lot of people listening here, spend a lot of time worrying about these two assumptions that sites change independently and that sites change identically, because we know that neither of those assumptions are actually true. And in fact, I would say that if you look at the literature, in fact, there's probably more people who spend time worrying about this assumption that sites change independently than the assumption that sites change identically. And sort of the focus of my talk is on this identical assumption. And one of the things that I would sort of contend to people listening is that in the field of phylogenetics, we probably spend too much time worrying about this independence assumption, which I think is a very hard thing to actually fix in any sort of tractable way, and not enough time worrying about this identical assumption, which is actually much easier to fix. And I think I will show you can get huge improvements by fixing that assumption. So just to sort of give a graphical illustration of what might be happening with a conventional phylogenetic substitution model, what I'm going to draw here, and I'm going to show a lot of plots like this, is the stationary state at each site of some substitution model. So we know that these substitution models are matrices. So for example, for an amino acid substitution model, it's a 20 by 20 matrix. And these matrices have some stationary state, which is a distribution that we would expect to see of every amino acid if this gene evolved for a very long time. And so, for example, if you take a standard Goldman-Yang model where you're estimating the codon frequencies empirically, and you apply that to influenza hemagglutinin, for this little stretch of a gene right here, these are the stationary states you would get. So at each site, there's some stationary state, and all these stationary states are the same across the sites. Now, if you actually go and you look at some alignment of influenza hemagglutinin sequences and look at the frequencies of the amino acids, you'll see something that looks like this. So this looks very different than that. And of course, not all of this is due to this substitution model at the top being incorrect, because the influenza sequences we observe in nature haven't evolved each independently for an infinite amount of time. So there's some historical contingency there. But even taking that into account, you can see the fact that some sites have changed a lot and some sites have not changed at all suggests that there's probably very different constraints on different sites in these genes. And in fact, there's a huge amount of protein biochemistry that indicates that that's true. So just for the particular example of influenza hemagglutinin, we know that this cysteine here and this cysteine here form a disulfide bond, and that's integrally important for the hemagglutinin protein to function. And so we have a biochemical reason why we think that these cysteines would never change. And that's not something that's in this substitution model. So there are people who have thought about this problem, and it's useful to think about why the problem is somewhat hard to fix. So although I described how the likelihood calculation frameworks can allow you to use substitution models like this, as soon as you assume that they are not identical across sites, so as soon as you make them make this statement no longer true, you start to have a lot of parameters you need to deal with. So in the Goldman-Yang model, we have a large number of sites in our gene, and we basically only have one set of equilibrium frequencies or stationary states we have to estimate, and then we apply that to every site. If instead we put a different stationary state for each site, as I've drawn here, the number of parameters required by this model grows a lot. So for example, for a 500 amino acid gene, there are 19 independent amino acid states at each site, because one of those states is sort of 1 minus the frequencies of all the other ones. So you have 500 times 19, so about 10,000 parameters that you would need to specify this site-specific substitution matrix. So it would be these 500 times 19 plus maybe a few other parameters at the nucleotide level. So nonetheless, papers have outlined what these site-specific substitution models would look like if we knew all these parameters. So this, I think, is a really important paper that was published by Halpern and Bruno in 1998. And they sort of looked at the scenario of if sites were changing independently and you knew these stationary states for each site, how would you calculate evolutionary distances? And they worked out some formulas related to this sort of treatment. And I think this is a very useful paper, but they didn't really deal with the question of how you got all of these 10,000 site-specific parameters. There's a really nice paper by Nicolas Rodrigue and Lartelet's group that develops a Bayesian method that actually does try to co-infer all of these stationary state parameters along with the phylogeny. And the reason that they do this in a Bayesian framework is if you put an appropriate prior over this, you don't have to worry about overfitting. And I think these methods are really nice and elegant, but they still face some problems. One is that they're slow because there's a lot of parameters that have to be sampled from. And another one is, even if these parameters are formally treated right, you only have sort of a finite amount of data. So your ability to sort of sample from them or to have enough data to really make your sampling from them capture these constraints may be limited. There's papers that have described maximum likelihood methods to infer these site-specific parameters from a phylogeny. So this is a really nice paper by Asif Tamuri that does this. And these are very powerful if you have a phylogeny and the question you want to ask is, what is the estimated effect of mutations? But in a maximum likelihood framework, you can't really take these parameters that you've inferred using this sort of method and put them back in your substitution model to describe the same data from which you got the parameters without having a lot of issues associated with overfitting. So, and obviously Tamuri is aware of that, and he presents this paper as not a way to get substitution models, but to estimate the distribution of selection coefficients. But I'm just bringing this up to point out why you can't just take these parameters directly from the same data you're analyzing in a maximum likelihood framework. There have been papers that have tried to computationally predict these site-specific constraints. So for example, they might use a three-dimensional model of a protein and try to estimate how each mutation would affect the function of the protein in the context of this model. And there have been a number of papers on this. This is one that I really like. And I think a nice thing about this paper is they show that these statistical potentials, so these potentials that are looking at the three-dimensional structure of the protein, have a little bit of power, but they're also pretty honest about their approach, and they critically evaluate how well it does or does not work, and sort of come to the conclusion that our ability to take a protein structure and model in any sort of tractable way the exact site-specific constraints is somewhat limited. And then another sort of nice advance that has been made is papers that just partition sites into a smaller number of predetermined substitution categories. So the advantage here is you're not estimating parameters for each site in the gene for these stationary states, but you somehow reduced those number of states, and in some cases you've predetermined their stationary states, and then you just assign sites within those. But again, we run into this problem. So, all right, I'm gonna pick back up on this talk until I hear otherwise. So I apologize if I'm repeating myself, and I also apologize if I've skipped a little bit, but what I said that I was going to present is how in specific cases where we have experimental data, we can build much better site-specific phylogenetic substitution models. And I first wanted to give an example. Eric, are you there?
Yeah, I am. I'm sorry. Yeah, just getting all set up. Sorry about the interruption.
Oh, no problem. Am I at the right place, or do I need to step back?
Yeah, this is perfect. So just keep going, and
Okay.
I'll pop in again if there's a problem.
Okay, so these measurements are made using a technique called deep mutational scanning, which was developed by Doug Fowler and Stan Fields at the University of Washington. And here's a nice review if you just want to read more about that general technique. And I'm going to describe just a specific example of deep mutational scanning that generates one of the data sets that I'm going to describe. And that's just so you can understand where the data that I'm showing and that I will be using comes from. So this is a paper we published that used this technique to measure the effects of all mutations to influenza hemagglutinin, although most of the next few slides I'm going to show are not directly from that paper because we've improved the experimental techniques somewhat. So the idea of a deep mutational scanning experiment works as shown here. You have some wild type viral gene, or gene of whatever you're studying. You make all of the mutants of this gene. You put them in your organism of interest, which in the case of influenza is an influenza virus. And you grow these viruses for a short period of time under a condition that puts selection on that gene. You then use deep sequencing to count all of the mutations that you had to begin with and to count all of the mutations that you have after the action of selection has worked. You analyze that data and you get out information which is presented here in a form that can go sort of directly into a phylogenetic substitution model. And so what you can think of these logo plots as representing as at each site, so here, for example, is site 190, if we had put in all of the 20 amino acids at equal frequency, how frequent would they have been after selection? So there's various ways you can sort of interpret this data. You can interpret it to have something to do with the fitness effect of that mutation. You can have something interpreted to have something to do with how well that mutation is tolerated. I've kind of used this slightly neutral term, which is the preference of that site for that amino acid, because I don't think it's quite right to call these exactly fitness values, because fitness is a property of not of a mutation, but of a gene sequence or an organism. And so what you can see is these experiments now allow you to estimate, at least in this particular genetic background, how preferred is each amino acid. And we might expect that to have something to do with how likely a substitution to that amino acid is to occur in nature. And this data is much different than just taking a natural sequence alignment and counting up the frequencies of amino acids. Because when you do that, the phylogeny, which is typically one of the things you care about, is one of the things that's affected what this distribution of amino acids look like. In the case of these experiments, we put in all of these amino acids. There's no phylogenetic relationship among these genes. We perform this selection, and we can sort of think of it as just drawing and then testing variants from this initial ensemble. So to just show a little bit more data, I don't want to get too bogged down in this, but I think it's important to understand the principle of these experiments. Here's a very kind of qualitative description of how these experiments work. We start with our gene that we care about, and we just deep sequence that gene to make sure that when we sequence that gene, we accurately return the wild type gene. And so here's the rate of mutation we see when we sequence that gene. And you can see that when we sequence it, we basically get back the wild type gene. And there's some tricks we use to make this deep sequencing accurate. I don't think they're that relevant to this talk. So I'm just going to scroll very quickly through them, and anyone who cares can go back to those slides and freeze them in the video and look at them, but I'm not going to explain them in detail here. Okay, so the upshot of that is that's how we're able to sequence this gene and get a low error rate. Then what we do is we take this gene and we mutagenize it to make all of the codon mutants of the gene. So both the single nucleotide changes, which are the immediate steps that would occur in evolution, and also the multinucleotide codon changes, which would probably require multiple steps to occur in natural evolution. And so when we look at our mutant genes, we now see single nucleotide changes to the codons, double nucleotide changes to the codons, and changes where all three nucleotides are altered, such as right here. And those introduce some stop codons, some synonymous mutations, and a lot of non-synonymous mutations. And sometimes people ask me, aren't the synonymous mutations supposed to be 25% of the mutations? And in fact, the nature of the genetic code is such that when you mutate at the nucleotide level, 25% of mutations are synonymous. But when you mutate at the codon level, a much smaller fraction are synonymous. Then we do more controls. So for example, we make wild type flu viruses from our wild type genes and sequence those and make sure we have a low error rate. And once we've done those controls, we can do the experiments we really care about, where we take these mutant genes and we use them to generate a whole bunch of mutant viruses, which we then deep sequence. And here's where you can see the thing that we care about, which is the action of selection on these mutations. So you can see clearly deleterious mutations, like stop codons, go away. Frequently neutral mutations, like synonymous mutations, stay at about the same frequency. And mutations which are sometimes deleterious, like non-synonymous mutations, decrease in frequency. So the nice thing about this data is not that you can observe these sort of qualitative overall trends, but that you have good statistics about what happened to each individual mutation. So there's about 30,000 unique codon mutations you can make to a 500 amino acid gene, and we might sequence at a depth of about 1 million. So in addition to these average statistics, we can see how each individual mutation changed. And that's how we get these so-called site-specific amino acid preferences. We analyze these statistics from this experiment, and we do that in the following way. We define the preference of site R for amino acid A to be this quantity that sums up to 1, just like the stationary state of any sort of substitution model has to sum up to 1. Then we say in our experiment, we mutated each site R to codon X at some rate prior to selection. Note that this mutation rate doesn't have anything to do with the mutation rate in natural evolution. This is the mutation rate that we put these things in our experiment. We then make an assumption, which is not a necessary assumption, but is one that we're making in our approach right now, that selection happens on amino acids. So we let A of X denote the amino acid encoded by codon X. And then we define these preferences as follows. The frequency of an amino acid after selection is the rate at which it was introduced initially times the preference for the amino acid that codon encodes, and then normalized so that all of these preferences sum up to 1. So once you've defined these preferences in that way, given the preferences, you can you have an estimated frequency for how much each mutation should occur. And in these actual experiments, the counts of that mutation should be multinomially distributed around these expected frequencies. So you can then calculate the likelihood of the deep sequencing data you got given these preferences and these mutation rates. And these mutation rates were measured by sequencing the DNA before you made mutation, or before you did selection. And you've also determined this quantity by deep sequencing. And so you can then place some priors over what you think these preferences look like. So we just start off assuming they're uniform across the gene. And then you can compute the posterior over these preferences and we do that, and then we summarize that by its mean. And so that gives us the data that's shown right here. This is saying in this deep sequencing experiment, when we put in all the mutations at equal frequency, how often do we see each of these mutations after selection? And each of these stacks represents 20 data points or 19 independent data points, and we have them for all 500 sites in the gene. And this sort of technique can be applied to lots of things in addition to influenza, as I've described. So you can also do this similar technique for cells, such as yeast or bacteria, which you can just put genes into and do functional selections on. And probably most of you have heard about all these new genome engineering techniques like CRISPR and Cas9. And so probably very soon people can also put these genes into human cells. So I would say in the future, there is going to be a lot of data of this type where people have measured the effects of all of the mutations to a gene. And so in the phylogenetic models that I'm going to talk about for the rest of this talk, I'm going to talk about using data from 4 published deep mutational scanning studies. One was done on a yeast protein, Gal4. One was done on beta-lactamase. And then 2 were done on influenza genes by my group. And so for those of you that say, how useful or generalizable are these substitution models you're talking about, I can tell you right now, you can use them for exactly 4 genes out there, which may not seem like very much, but as I suggested on the previous slide, I think that if you go back and look at the same list in a few more years, it's going to be much, much longer. Okay, so how do we put this deep mutational scanning data into a quantitative model? So we go back to the beginning of the talk where we talked about how these substitution models are these stochastic matrices which give the probability of changing from identity X, and we're going to talk about codon models, so that's a codon, to identity Y, and we're going to have a different stochastic matrix for each site. So the first thing we do is a fairly standard thing in what is sometimes called mutation fixation or substitution models, where you imagine decomposing this matrix into 2 terms, one of which represents the rate at which these mutations occur, and the other one that represents what tends to happen to these mutations after they occur. So in the models that I'm going to talk about, we're going to assume that this mutation process is uniform across the gene. So it is not site-specific. It's identical across sites. And we're going to model it in a pretty standard way, which is an HKY85 model, which says that there's some equilibrium frequencies for the 4 different nucleotides, and that there's some distinct rate of transition mutations versus transversion mutations. So sorry, another typo here. This should say transitions versus transversions. And this assumption is obviously not completely correct because we know that mutation rates differ across genes, but at least for the type of genes that I'm talking about, it's probably not that bad of an assumption to say that mutation is quasi-uniform across the gene. Sort of the interesting part of what I'm going to describe, and the only part that is novel compared to what other people have done, comes in this selection term here. And this is where we put in this deep mutational scanning data. And so what we want to do qualitatively is we want to say mutations from preferred amino acids to other preferred amino acids, such as from serine to threonine, you might think those will be fairly likely to fix. And mutations from highly preferred amino acids, such as cysteine here, to not very preferred amino acids, such as tyrosine here, are going to be very unlikely to fix, and mutations from tyrosine to cysteine would be very likely to fix. So this is where all the site-specific information comes in. And we do that parameterization using this framework that was developed by Halpern and Bruno in their 1998 paper. So I would that paper was mentioned earlier in this talk, and I'd refer you to that paper if you really want the details. I'll also point out an unfortunate fact. If you go through their paper, they actually end up having a typo in the key equation. So just be aware that's why this equation looks a little bit different than their equation. But they derive how, if these preferences were related to fitness effects of mutations, which again is not probably exactly the right way to think about them, what would be the fixation probability of a mutation going from some amino acid to some other amino acid. So when we write this equation, we say that, all right, if the amino acid doesn't change, there's some normalized fixation probability of 1 that the amino acid will fix. If the amino acid does change, then we put in some parameter which does not have a real sort of population genetics meaning, but which is some kind of phenomenological relative non-synonymous synonymous rate omega. And then we put in these experimentally measured effects of these mutations, these pi values. And then we put in another parameter here, which I've called beta, which is the stringency parameter. So a beta value greater than 1 sort of amplifies the relative effect of our measurements. So it means that if we measured cysteine to be favorable, we think that natural evolution thinks it's even more favorable. And a stringency parameter of less than 1 sort of flattens these things. So in the limiting case where this is 0, all amino acids are equivalent. And again, within the assumptions of this Halpern and Bruno paper, this beta term can actually be given some population genetics, meaning having to do with effective population size. I'm not really going to go into that further. There's also a nice paper by Dave McCandlish and Arlen Stoltzfus that talks a lot about how what the parameters in these sorts of models might mean. Okay, so now let's compare these models to a more standard model. So what I've written here is now a Goldman-Yang substitution model. So in a Goldman-Yang substitution model, you imagine that there's some baseline rate of substitution that happens for synonymous transversions. And then that rate is basically just proportional to the equilibrium frequency of the amino acid that's encoded by that transversion. You imagine there's some distinct rate of transitions versus transversions. And then you imagine that when a mutation changes the amino acid, it's retarded or accelerated by some parameter omega called the dn/ds value. And note that none of this depends on site R. So in a Goldman-Yang model, you're assuming that all sites have identical equilibrium codon frequencies. So as compared to the deep mutational scanning models, which have this site-specific information, the Goldman-Yang model would in the end be saying, again, that the constraints on your gene look like this. Okay, so what happens if we now compare a Goldman-Yang model to these experimentally informed models? So I described to you how there are 4 data sets that we can look at. One of them is beta-lactamase. And so here, if we use these experimentally informed codon models, we get some likelihood of the data given our model. And if we use the Goldman-Yang model that's called the M0 formulas, and if we use a Goldman-Yang model, so the version classified as M0 in this Yang, Nielsen, Goldman, and Krebs-Peterson paper, you get some other likelihood, which is substantially worse. And that's even though the experimentally informed codon model has many fewer parameters that are determined from the sequences that you're analyzing. And again, the deep mutational scanning data is something that's measured independently in an experiment, so it's not really fit from the sequences you're analyzing. You can also look at the values of the specific parameters. So in a Goldman-Yang style model, you fit an omega, so a dn/ds ratio that's quite a bit less than 1, which suggests that non-synonymous mutations are disfavored. When you use these experimentally informed models, which are actually already accounting for the fact that different sites prefer different amino acids, and for which the omega parameter only accounts for the additional acceleration or retardation of that substitution rate after accounting for those preferences, you see that when you account for the protein level site-specific constraints, the amino acids are evolving at the same, amino acid substitutions are fixing at exactly the rate you'd expect. We can also fit this stringency parameter, and we see in this case it's right about 1. So the experiments seem to be capturing the constraints just about right. Here's another example. So this is for influenza nucleoprotein. Again, the experimentally informed codon model describes the data much, much better than the Goldman-Yang model. Again, the omega value that's fit from the nucleoprotein data set is a lot higher than the one fit from the Goldman-Yang model, which again suggests that most of the slowdown in non-synonymous substitution rate is captured by these site-specific constraints. And here we fit a beta or a stringency parameter much greater than 1, which indicates that natural evolution is favoring the same things that are favored in our experiments, but with a much higher stringency.
Does that surprise you, Jesse? I mean, like, this... Doesn't... I mean, I would think that beta would generally be less than 1.
So the reason I would think that it would be greater than 1 is in our experiments, I would think that the strength of selection in nature is a lot stronger than what we put on in our experiments. So for one thing, we know in our experiments there's always some rate that non-functional viruses will kind of leak through due to genetic complementation from other viruses. And in general, I think that natural evolution generally imposes much stronger pressures than people do in experiments. So for example, when people will experimentally make measurements to mutational effects to proteins, they'll find a lot of mutations that look just about neutral in the experiment, but yet those mutations don't fix in nature. So I actually think that natural selection tends to be a lot more discerning than most of the experiments we do in the lab. So that would be my argument anyway, why it's the case. Although, as you'll see, it's not the case in every example. And again, you would expect this beta to have some connection really to the difference between the effective population size in nature and the effective population size in your experiment. And certainly in our experiments, it's not really very easy to estimate what would be the equivalent of the effective population size.
Well, I mean, if we were to extrapolate from these 2 points, I mean, maybe the leak through is less in the beta lactamase experiments than it is in the flu proteins, because there's less probability of getting the leak through on the virus.
Yeah, I think that's probably true. So due to sort of technical aspects of how these experiments are done, the bacteria there's less chance for non-functional variants or marginally functional variants to squeak through in the bacterial selection than there is in the influenza selection. So that's at least one of the possibilities. When we look at influenza hemagglutinin, we see a similar trend that a lot of the slowdown in non-synonymous rate that's determined by a Goldman-Yang model actually is just accounted for by these site-specific constraints. So omega gets a lot closer to 1. And again, the stringency of selection in nature is higher than the stringency of selection in our experiments. And when we look at this GAL4 data set, we see the same trend for omega, although it's much less pronounced. And this is the one example where the stringency of selection in nature turns out to be different than the stringency, or turns out to be less than the stringency of selection in the experiments. Although it's also important to remember that these beta parameters really only compare the stringency of the selections if the selection pressures are also the same. So it's also possible that the experiment has different selection pressures and the beta is a little bit less than 1 to partially flatten out inaccurate measurements.
So this is kind of expected that these are ones that you're getting?
So I would say, yeah, so the question that David just asked, David's one of the people in the room, is it expected that the Goldman-Yang omegas would be less than the omegas that you get from the experiments? And I think the answer is yes. So if you go back, and sorry, I'm going to have to scroll back a few slides here, but if you go back to a Goldman-Yang model, you're basically assuming that there's almost no site-specific amino acid constraints because the stationary states are all the same. So in effect, in a Goldman-Yang model, let's say that this particular site here always stays at valine during evolution in nature because anything else is disfavored. Because a Goldman-Yang model doesn't have any way to make that preference for that valine very high, the only way that it can sort of keep that amino acid at valine is to pretend that any sort of non-synonymous mutation is disfavored at that site. So versus in the experimental data, which I'll show here, you can imagine that you don't have to you can keep this site as histidine not just by shrinking omega, but just by taking account of the fact that your stationary state now says this site will tend to remain histidine. So that's how I would kind of explain that. So to go back to the stringency parameter point, again, I think this is sort of an interesting and sort of potentially untapped area that certainly I don't have the population genetics expertise to tap, but this parameter does mean something under certain assumptions, and certainly we can see that if we just look at our data, here's what the data for hemagglutinin look like coming out of our experiments, and if we rescale it by the stringency parameter, which is in effect the equivalent of raising each of these proportions here to the power, in the case of hemagglutinin, 1.6, and then renormalizing them to sum to 1, the data goes to looking like that. So you can see that this is saying in natural evolution, so in our experiments, histidine is favored, but some other positively charged amino acids sometimes get through. In nature, histidine is favored and pretty much nothing else ever gets through. So this is sort of the idea that nature is favoring the same thing as in these experiments, but favoring it more stringently in the case of most of those genes. So the last thing that I want to talk about is, all right, beyond sort of fitting phylogenetic models, can we actually do anything useful with this? What I'd like to talk about is the idea of detecting positive selection. So detecting positive selection really comes to the idea that you'd like to find sites and genes that are evolving differently than some null model of how evolution should happen in the absence of positive selection. So a really powerful approach that was developed about 20 years, a little over 20 years ago to do this is so-called DNDS approaches. So what those approaches will typically do is they'll take a model like the Goldman-Yang model, and they'll fit a phylogeny, and they'll also fit all of these parameters that describe the phylogeny. And that will be sort of the baseline that you'll start with. And then what they'll do is, keeping everything else fixed, they'll go and they'll fit a different synonymous rate and a different non-synonymous synonymous ratio to each site. And then you can test the alternative hypothesis that this non-synonymous synonymous ratio is not equal to 1 to the nested null hypothesis that it's equal to 1. And if there's evidence that this ratio is greater than 1, you would say the site's under positive selection. So actually selection is favoring amino acid changes. And if it's less than 1, you'd say the site is under purifying selection. So natural evolution is disfavoring amino acid changes. And this is one particular sort of instantiation of this approach, the FEL approach, at least as it's implemented in Sergei Kozakovsky-Pan's software. And there's a lot of sort of other variations of the same test that do slightly different things, but in the end, they're sort of qualitatively all doing the same thing. They're trying to find sites that are changing faster or slower than expected. And some of the advances over this model that allow things for like looking at branch-specific effects and things like that are actually very powerful advancements. So I'm not going to talk about those, but they're very important. But qualitatively, in the end, the thing that's always being tested is a site evolving at a rate that's equal to 1 or not equal to 1. Oops, sorry. Okay. And then, so unsurprisingly, under this null model, if you typically look at a gene, you see that a lot of the sites are evolved at an omega value less than 1 and are deemed under purifying selection. And so to go to David's question, sort of, is this an interesting finding? And I would say it's not really an interesting finding if you believe that your gene is evolving like anything other than a random nucleotide sequence. So we already know that for pretty much every protein, there's a lot of constraint on the amino acid sequence. So you'd expect most sites to not just be flipping sort of with between random amino acids at some rate, at the same rate as their underlying mutation rate. Sorry, I keep treating this as a PowerPoint presentation. Okay, and so the fundamental problem of this approach, if we go back to looking at it like this, is our null model has assumed that the constraints are equivalent on every site. So the only thing that we really have power to identify is a site that's changing much, much faster than other sites around it, and much, much faster than you would expect. So now if we go to the case where we have more experimental data, you can imagine cases where you'd like to be able to tell other kinds of mutations apart. So this is, for an example, from experimental work my group has done on influenza nucleoprotein, and I'm not going to go into details of all the experiments, but one of the things that we found is there were 2 different mutations that occurred in this gene at around the same time. One of these mutations was M136I, and as far as we can tell, that mutation has no real biological significance. And if you look at the deep mutational scanning data we have for that gene, you would think that this is not a very surprising mutation, because methionine and isoleucine are both highly preferred amino acids at site 136, and you shouldn't be that surprised if there's a substitution from one to the other, just like you wouldn't be that surprised if there was a synonymous substitution at that site. Another mutation, which there's a lot of experimental evidence from our group and for other groups, it's very important for helping influenza escape from the immune response, the T cell arm of the immune response, is a change of site 384 from this highly preferred amino acid arginine to glycine, which is so small here you can't even see it. So given this deep mutational scanning data, you would expect that this is a pretty surprising mutation. It indicates some process that's going on in nature which is different than the null model that's established by these deep mutational scanning experiments. So we'd like to be able to identify that kind of selection. So how do we do that? We do something very similar to these DNDS approaches, but we use the deep mutational scanning data to do the comparison with a much higher level of subtlety. So again, we know all of this stuff about different sites, so we can look at amino acid mutations and say, are they surprising, or do they look like they're basically going to be the functional equivalent of synonymous mutations? And to do that, we can do two things. First, we can detect what you might call diversifying selection, which are just sites that are changing a lot faster than you would expect. And so you do something exactly equivalent to this FEL approach in the Goldman-Yang model, but you do it now where you've written this experimentally informed codon model. So for each site, you fit a site-specific synonymous rate and a site-specific non-synonymous rate, and you test if that non-synonymous rate is greater or less than 1, if there's evidence for it being greater or less than 1. So what happens when you actually do these genes? First, let's look at this Gal4 gene, which we have this big data set for. So Gal4 is a conserved gene in yeast. We think that what we measure in the lab captures what this gene does in nature, and we think the function of this gene has been conserved for a long time. So if you use a Goldman-Yang type model and you ask, are sites under so-called positive selection, so this DN-DS ratio is greater than 1, or so-called negative selection, where it's less than 1, what you find is for a large fraction of sites at a false discovery rate of 0.05, all of the sites that are under this dotted line here would be deemed under purifying selection. So they'd have omega R greater than 1. On the other hand, when you use this experimentally informed codon model, you find that at a false discovery rate of 0.05, so these dashed lines indicate what the p-value would have to be for a site to not be deemed having omega equal to 1, you find that all of the sites are pretty much evolving as you would expect. So here's an example, to go to David's question, of why the experimentally informed models tend to give you larger omega values, because they're already accounting for these constraints which are slowing down the rate of substitution at some sites. And so, for example, the site that are way down here in the Goldman-Yang model, they're still changing slowly under the experimentally informed model. It's the same data set, but because these site-specific constraints tell you you expect the site to change slowly, you don't estimate this parameter as being way down here. Sorry, again, I'm scrolling the wrong way. You can do the same thing for the lactamase gene. So again, under the Goldman-Yang model, you find that no sites are changing faster than you'd expect at that level of statistical significance, but a small fraction are changing slower. When you use the experimentally informed models, you instead find that a small fraction of sites are changing faster than you'd expect. You see something similar for nucleoprotein, again. So the experimentally informed codon model is telling you that no sites are changing slower than you'd expect, but a few are changing faster. The Goldman-Yang model tells you the opposite. And you see the same thing for influenza hemagglutinin. Another really nice thing you can do with these experimentally informed substitution models is you can find a different kind of selection that's pretty hard to find with the Goldman-Yang model. So another form of biologically interesting selection is a site that's not changing faster than you'd expect, but is just changing to different identities than you would expect. So we have these experimentally measured amino acid preferences for each site. And you can now fit a parameter which says, in nature, is that site changing to something different? So is there a different preference for that amino acid in nature than there is in your experiment? So there are now, for each site, there are 19 of these parameters here. They have to sum up to 0, because anytime natural evolution is favoring one site more, it must be favoring some other site less. And there are now 19 of these parameters, and we can fit them all with some sort of regularization. So I don't currently have a good method for actually doing significance testing on whether there's evidence for these things being different than 0, but at least we can fit them under some sort of regularization term. And when we do that, we can now say, are there preferences for unexpected amino acids? So for Gal4, most of the sites are pretty much substituting to the amino acids that you think they would. So the y-axis here is the magnitude, the total magnitude of preferences for unexpected amino acids at a site, and that could go as high as 1. And for Gal4, most substitutions are favoring the amino acids that you would expect. For lactamase, most sites are favoring the ones you expect, but there's a few sites that are changing to very different things. Same thing for influenza nucleoprotein, and same things for influenza hemagglutinin. And another way to look at that kind of data is as shown here. We have from our deep mutational scanning some expectation of which sites should be favored in nature. And at certain positions, so for example, here at position 189, we see that natural evolution is favoring something different than we expect. So this representation here is showing you that at site 189, you expect natural evolution to favor one of these negatively charged amino acids. But when you look, there's a decreased preference for the negatively charged amino acids and an increased preference for the positively charged amino acid arginine. And so this is a very different kind of selection than the diversifying selection captured by DNDS. It's not a site changing faster than you'd expect, it's just that it's substituting to something different. So this suggests that the selection pressures in your experiment and the selection pressures in nature are different. So I'd just kind of like to end with an anecdotal example from the beta-lactamase gene, which I think illustrates some of the power of this approach. So really, when we talk about trying to identify selection in genes, what we really want to do is we want to identify mutations that have some biological significance. So beta-lactamase is an antibiotic resistance gene, and it's under pressure to get mutations that help it escape from new antibiotics. And there's been a lot of work identifying what the mutations are that help it escape from new antibiotics. So I'm showing here 5 different sites in this gene, 3 of which, 238, 240, and 244, are known to help lactamase escape from antibiotics, and 2 of which, 201 and 242, have no known role in antibiotic resistance. So what happens when you look at these sites? At site 201, you see some mild variation. At site 238, you see some substitutions. At site 240, you see some substitutions. At site 242, it's completely conserved. And at site 244, you see some substitutions. So if we didn't have any of this experimental data, all we could do is take these naturally evolving lactamases and fit a Goldman-Yang model and estimate the site-specific omega. If we did that, we'd see that site 242 is changing really slowly, so it's down here under purifying selection, and none of the other sites are under positive selection at a false discovery rate of 0.05. But now we have this experimental data. So our experimental data tells us that site 201, it's changing in nature, and we should basically expect it to be changing because it's highly tolerant of a lot of mutations. If we look at site 238, we see that in nature, we expect this thing to be glycine or possibly alanine based on our experiments. So the fact that it's changing from glycine to serine is very surprising. It indicates that something's happening in nature that's not there in our experiments. At site 240, we can see that it favors a lot of different amino acids, but there's no particular preference for lysine. So the fact that it's repeatedly changing to lysine in nature might be deemed sort of surprising. Site 242, we see that it's completely conserved as nature as glycine, and that's pretty much what we expect. In site 244, we see that in nature we expect it to remain arginine, so it's surprising that it's changing to a lot of other things. So now when we use these experimentally informed codon models, what we see is 2 of these 3 resistant sites, site 244 and site 238, are actually changing faster than you would expect when you account for the fact that they're actually under a substantial amount of constraint, as shown here and here. So we now have power to detect this diversifying selection that you couldn't detect with a Goldman-Yang model. Furthermore, when we look our sites changing to unexpected amino acids, we see that we can also now flag site 240, and that site is surprising because it keeps changing to lysine. So it prefers lysine in nature, even though clearly it doesn't need that lysine under the selection conditions in the lab, which obviously does not involve people continually using new antibiotics. And again, just to kind of beat a dead horse, the reason that we're able to detect the selection much better is we now have these really good null models for how these sites should change rather than these rather crude Goldman-Yang equilibrium frequencies. Sorry, again, I'm scrolling through this the wrong way. So to just sort of summarize, I guess if you were to try to encapsulate everything I've said in a graphical form, before we did not have a good idea for how a gene, for example, an influenza gene, should evolve. So for example, in a Goldman-Yang model, we're forced to assume something like this, where the constraints are uniform across the sites. Now we do these experiments that allow us to experimentally measure the constraints at each site in the gene, so we can see that there are highly non-uniform constraints, and we really expect different sites in these genes to be evolving differently, as indicated here. And then if we look at genes in nature, we can see that they are in fact evolving non-uniformly. And in fact, most of the ways that they're evolving are pretty similar to what we're measuring in our experiments. So that means both that our experiments can allow us to build better substitution models, and in those rare cases where there's deviations between our experiments and what happens in nature, we can posit that there's some sort of interesting form of positive selection going on there. And so really the job of phylogenetics, in an area that I think there's a lot of room for continued advancement in, is how do we use these sources of experimental data and other sources of high throughput information to really create models that can be used to analyze what's going on in nature. And so I'd like to wrap up by thanking the people in my group who contributed to the work I'm talking about. Bhargavi was a former postdoc in my group, or is a current postdoc, and Mike is a current postdoc. And then I'd also just like to mention some of the references from our work that are useful if you want to read more about these. And I put them in chronological order. You should probably read the last one first if you're only going to read one of them. But these are examples of where we used our own deep mutational scanning data or other deep mutational scanning data to try to build these phylogenetic substitution models. Okay, thank you.
Thank you very much, Jesse. That was, of course, really amazing talk. Maybe I'll start off the questions by asking, like, does the constancy of the beta parameter across sites bother you at all? I mean, it just seems sort of funny that we have this beta parameter that we have to fit, but then we assume that it's the same across sites. And I know that we can't have like a one beta for each site, but I wondered if what would happen if you fit a two-component mixture or something like that.
Yeah, that's a great. So I'm going to just scroll back a little bit to get to a slide that'll help me answer that question. So Eric's question has to do with the stringency parameter beta, which corresponds to taking your experimental data and transforming it to the selection being stronger or weaker.
Sorry, the medium
yeah, we'll be done before then. And so as Eric was pointing out, you're going to have different selection at certain sites in nature than the experiments. So in terms of fitting an overall model, one way you could do that is you could, rather than fit a single beta parameter, you could do what's called fitting a mixture of two beta parameters, just like people fit a mixture of multiple omegas in a Goldman-Yang model. We haven't tried that, mostly just because we don't know how to get the computations working. But something that we have done when we were trying to identify what I've called differential selection is, rather than fitting this vector of 19 differential preferences at each site, so saying how much is it changing, you can just fit a site-specific stringency parameter, just like we fit a site-specific omega parameter. And then what will happen is, if a site is evolving more uniformly in nature than in the experiment, you'll fit beta to 0. If it's evolving under a much stronger constraint in nature than your experiments, but the same constraints, you'll fit a beta of infinity or larger than 1 anyway. The only downside of that approach, and the reason we have, and the nice thing about that approach is you can also then calculate p-values using a likelihood ratio test. The downside of that approach is that fitting the stringency parameter only allows for two possibilities. Either your experiments measured the right constraints, they just didn't measure them strong enough, or natural evolution is more permissive to all amino acids than you measure in your experiment. And the advantage of fitting a different preference to each site, although it doesn't allow you to make good statistical hypothesis testing, at least allows you to accommodate the possibility that there's constraints in your experiment and constraints in nature, and they're different. So that's the best answer I can give to that, I think.
Yeah, that's a satisfactory answer for sure. I mean, right, and then if you are fitting these per site betas, then you also can't do this nice omega. I mean, you sort of absorbed all of your information into these per site betas, and you can't do the omega on top of that.
Yeah, I think you're correct in that. But I do think fitting some of these things for, I mean, I would say almost all of the, I'm talking about in this talk really kind of one of the simplest versions of the Goldman-Yang model, and all of these, tremendous improvements that have been made to the Goldman-Yang model, like allowing omega parameters to come from gamma distributions and fitting, branch specific things, and all of that stuff is really good, and would probably, I think someone with sufficient computational skill anyway, which is probably not me, could also be applied to these experimentally informed codon models. So I think that your suggestion is really a good one. It's just something that we haven't done.
So, I mean, what's next? I mean, like, obviously you've made a lot of progress using these per site models. I mean, is it just scaling things up to lots of different genes, or?
Yeah,
like...
I mean, so what I would say anyway is, I mean, as you know, I'm sort of someone who's interested in phylogenetics and quasi-competent in phylogenetics, but a lot of what my lab does is these sort of experimental analyses. And my belief anyway is that these experimentally informed models, there's going to be more and more data like this, and this data can be used in models like I'm talking about here in more sophisticated versions. So I mean, I guess my hope for the phylogenetics is that my group will keep plugging away on this until or if we convince someone who's more competent at phylogenetics and writes some of these really good phylogenetics software packages to get interested in it. And hopefully, I would hope that in 5 or 10 years, when you're choosing your substitution models, there might be an option for uploading your site-specific deep mutational scanning data and using that in the substitution model if you're trying to fit a phylogeny or trying to detect selection. So that's kind of where I hope it goes from the phylogenetics perspective. From the conceptual perspective, I think the big question, and one that I did not talk about as much here, is to what degree do these site-specific amino acid preferences themselves actually change during evolution? So we know that there's epistasis among sites. And so in principle, you could imagine that after a while, for example, this gene no longer requires histidine at site 183. It wants to have some other identity. And so there was a nice paper by Pollock and Goldstein that talked about this phenomenon, calling it like a Stokes shift or a shift in amino acid preferences. And I think those things definitely happen. I disagree with Goldstein and Pollock in terms of how common these shifts are going to be. I think based on both some direct experimental evidence and some fitting of these sorts of models to highly divergent genes, that there's a lot of evidence that these amino acid preferences are more conserved across broad sets of homologs than a lot of people appreciate in phylogenetics. But I do think that they definitely shift to some degree. So one of the projects that my group is most interested in is taking a series of increasingly diverged homologs of the same gene and measuring these site-specific amino acid preferences so we can see how much they change as the gene evolves. So it's not immediately obvious how that new information would be put into a better phylogenetic model, because for reasons I discussed, once you start putting in interactions between sites, which are really what makes these preferences shift, a lot of the whole machinery of phylogenetics starts to break down. But I think by at least bounding that phenomenon, so figuring out how big these shifts actually are for real proteins, and also understanding their properties, maybe we can eventually start to come up with models that will also account for the fact that occasionally the constraints on a gene are shifting as the gene evolves.
Yeah, very interesting.
David, you had one question?
Yeah, so with such like this consistent negative bias in this Goldman-Yang model, is there any kind of idea built into it that you could do a sort of bias correction using these experimentally informed data? So you have like all of this prior knowledge that it seemed like maybe you weren't using for the Goldman-Yang.
Yeah, so this is a great question. So, and I'm not sure if people listening to this can hear this. So what David's question was, and correct me if I'm not phrasing it correctly, but the Goldman-Yang model really only has this single parameter omega, which can sort of account for selection on amino acids by retarding your substitution rate. And so is there some way to augment that Goldman-Yang model with this sort of information so you could keep the basic framework of the Goldman-Yang model, but slow down the substitution of sites based on what you would observe in these experiments? And so I think that can be done. There's a really nice paper by Stephanie Spielman and Klaus Wilke that was published, I believe, last year in Molecular Biology and Evolution. And there what they do is they say, if you knew what these site-specific parameters are, what would the omega end up being for that site? So like how much would the non-synonymous substitution rate be slowed down. So they're sort of connecting these two things. What does omega mean in the framework where there are these sorts of constraints? And I think it's a really cool relationship. So for the specific work I am thinking about, like where you actually have this data, I would say rather than kind of trying to augment or correct the Goldman-Yang model, you're probably better off just throwing away the Goldman, or you're not really throwing away, but you're changing the framework a little bit to make it site-specific and throwing away the whole non-site-specific nature of it. But I think there's a lot of, in a lot of cases, you still don't have this experimental data, and or you have analyses you can only do with the Goldman-Yang model. And in those cases, I think this sort of framework from the Spielman and Wilkie paper is super nice, because you can, for example, start to look at site-specific omegas and say, what does that tell you about what the distribution of these sorts of effects might look like for a lot of other genes. So I think that's one of the things you could do by correcting those things. And the next philo seminar I know is given by Klaus Wilkie. So I don't know what he's going to talk about, but maybe he'll talk a little bit about that aspect. But there is now, and this was only within the last year that they published this, some sort of formalized relationship between the omegas in a Goldman-Yang model and sort of site-specific parameters.
Very nice. So we should probably wrap up, but just very briefly, you used to work on protein structure. You know people who work on protein structure. Have you tried to come up with any sort of interpretation, like mapping these site-specific preferences onto some structural variants?
Yeah, I mean, so people have there are definitely people doing that. We're thinking about it a little bit. And what I would just say that it kind of emphasizes is when you look at these preferences and you know a lot about the specific protein, you can often rationalize them. So you can rationalize for hemagglutinin, I could tell you why this site has to be histidine because it binds to the receptor. But I think another thing that these measurements emphasize is why it's so hard to really go from structure to phylogenetic substitution models. So one of the things that people try to do, for example, if they're making more nuanced substitution models, is they would say, if a site is in the core of the protein, it should prefer hydrophobic amino acids. And we see in our data that's true. So site 178 and 179 are both in the core of the protein, and they both prefer hydrophobic amino acids. But how come this one likes leucine and this other one likes valine? That kind of stuff is extremely hard to rationalize. So I think these data both sort of illustrate kind of the broad trends that can easily be rationalized about proteins that explain maybe 50% of this constraints, and also these highly idiosyncratic things which are almost impossible to predict and sometimes almost impossible to rationalize even when you know what they are.
Nothing like a good experiment. All right.
Okay.
Thank you very much, Jesse. And yeah, so I guess we're all looking forward to Klaus's talk April 20th at 10 a.m. Pacific Daylight Time. Thanks again, Jesse, and see you next time.
Okay, thank you, Eric.